Okay, so we're going to talk about respiratory system physiology. I'm doing a little video of myself because I want to go over breathing with you guys so that you understand it since we're not meeting face to face. Oops. Okay, so functions of the respiratory system, obviously to provide extensive uh, gas exchange surface area between air and circulating blood. The goal of the respiratory system is to filter, warm, and humidify air as it goes down, and then move air to and from exchange surfaces of the lungs, protect respiratory surfaces from outside environment, provide speech, and aids in pH regulation by getting rid of CO2. Cells require oxygen for cellular metabolism, maintenance, growth, defense, and division, right? Carbon dioxide is a byproduct of cellular metabolism, and so your cells need to remove that CO2 and take in oxygen. Remember that we talked about this when we covered blood. Anytime you have CO2 in plasma, you're going to make carbonic acid. And so when those tissues produce CO2, they make carbonic acid. And that carbonic acid is going to dissociate into protons and bicarbonate ion. Protons are going to acidify that area. Your tissues also produce protons because of metabolism. We'll get into it a little bit more, but keep in mind that bicarbonate ion is a buffer, right? And so the goal of a buffer is to grab protons and maintain the pH of the plasma. So the respiratory system and the circulatory system work in concert to regulate pH of blood with kidneys and to get oxygen to the tissues and CO2 up. We can skip this one. We talked about it in the anatomy slide. The gas exchange area is the alveoli, right? And so the alveoli, one is an alveolus. So the alveoli are cluster-like sacs. They look like grapes, clusters of grapes, and they have epithelial cells that make up the alveolar sac. So these are squamosal epithelials. This is simple squamous epithelium. And then the capillary is actually simple squamous epithelium or endothelium. So the oxygen only has to go through 2 layers of cells, and CO2 only has to go through 2 layers of cells. The act of breathing. So pulmonary ventilation relies on gas traveling from high to low pressure. Boyle's law states that pressure is inversely related to volume. All right. So what we need to do to get air to move in is we need to make the volume or the space bigger so that pressure will drop so that air will go into the lungs. If you remember, so your first rule is that gas is going to move from high pressure to low pressure. So if you want air to move in to the lungs, we're going to make the lungs the low pressure area, right? How do we do that? We're going to make the space bigger. Okay. Inspiration is air moving in, right? Expiration is air moving out. So inspiration is based off of muscles contracting. So let's go through this. So your diaphragm and your external intercostals, this should be external, intercostals contract, okay? Let's talk about how they contract. So the diaphragm sits, I'm gonna do a sideways version. The diaphragm is right below your lungs, and the diaphragm actually contracts. And when it contracts down, okay? When it relaxes up. So diaphragm contracts down, your external intercostals between your ribs are going to contract. And when they contract, they're gonna pull the ribs up and out. And that is going to increase the volume of the thoracic cavity. And so pressure drops. When volume increases, pressure drops. So if pressure drops in the lungs, then air is going to move from the external environment into the lungs. Okay, that's how we actually take a breath. How do we exhale? So exhalation, normal exhalation is passive. all right? Why is it passive? Because these muscles relax because the nervous system stimulation stops. And so what ends up happening is the diaphragm relaxes up, the external intercostals relax down. And so the volume of that thoracic cavity gets smaller. And when that space gets smaller, pressure goes up. And when pressure goes up, air is going to move from high pressure, which is the lungs, to outside, which is the lower pressure. Okay? So contraction of your diaphragm, your external intercostals, space gets bigger, pressure drops, air moves in. Then these muscles relax, so your nervous system stops. Nervous system stimulation stops, so muscles relax and diaphragm relaxes up, and the space gets smaller. So space gets smaller, pressure goes up, right? And air moves out. Good. Here's a picture of this. So for inhalation, that diaphragm contracts down, the external intercostals are going to pull the ribs up and out, making that space bigger. Okay. Volume's bigger. What happens to pressure? Pressure drops, right? Let's see. So pressure drops. So we have a decrease in pressure, sorry, right? And so which way is air going to go? Air is going to go into the lungs. Whoops, sorry, you guys. My pen is going a little crazy. When your diaphragm relaxes and your external intercostals relax, they make the space smaller. So smaller space, smaller volume, right, of space, what happens to pressure? Pressure goes up, and pressure is lower here. So which way does air go? Air is going to go from here out, right? Good. Okay, so I'm going to pause for a second, and I'll be right back with you. Okay. So here's a side-by-side comparison. This is the end of inspiration. So the diaphragm has contracted, your external intercostals have contracted, you know, the accessory muscles when you're taking a deep breath would be your scalenes, your sternocleidomastoid, right? And then, or sorry, your pecs, your scalenes, and your sternocleidomastoid. When you are exhaling, it's passive, so those muscles just shut off. But let's say you were forcing a breath out. When you're forcing a breath out, you're going to use your internal intercostals and your abdominal muscles. Okay, so modes of breathing, we can classify modes of breathing by pattern of muscle activity or into quiet breathing and forced breathing. So let's go over quiet breathing. Quiet breathing is called eupnea. It involves active inhalation, right? And passive exhalation. What does that mean? Passive means there's no skeletal muscles contracting. So skeletal muscles are contracting for inhalation. They're not. They're relaxing for exhalation. Diaphragmatic breathing or deep breathing is actually dominated by the diaphragm. And then coastal breathing or shallow breathing is dominated by rib cage movements. Usually you can tell if somebody's diaphragmatic breathing or deep breathing because this is considered to be belly breathing. When somebody is trying to take deep breaths to relax, they're going to do deep breathing. Coastal breathing or shallow breathing, you can tell because if somebody is shallowly breathing or stressed out, their shoulders are going to move up and down more so. This is usually the type of breathing that people do when they're stressed out. And to decrease that stress response, you can actually shut it off by doing deep breathing. Forced breathing is called hyperpnea. It involves active inhalation and active exhalation. So you are contracting muscles to move air in. You are contracting muscles to get air out, right? You're assisted by those accessory muscles. So all of those muscles in your thoracic region are contracting to get you to ventilate. You can imagine that that's really exhausting, and it's really taxing to get maximum ventilation when you are force breathing. Pulmonary ventilation. So the respiratory rates and volumes. Your respiratory rate, or excuse me, your respiratory system adapts to changing oxygen demands by varying the number of breaths per minute, so your respiratory rate, and the volume of air moved per breath. Um, this depends on what activity you're doing. So normal respiratory rate, right, is about 60 on 60, about 12 to 20 breaths per minute, right? And the average volume of air that a person moves is around 500 mils. And so think about if you were running, how would that change? Right, so respiratory rate would increase, and your tidal volume would increase. Okay, so the volume of air moved per breath is your tidal volume. This is something that we would have done in lab. I want to go over this with you guys. Let me explain it first. So let me put on the laser pointer first so that I can go through this with you. And then I'm going to switch to do a little video. And then I want to draw it. And I want you guys to draw this out. So draw it out a couple of times so you get an idea of how this works, since we're not actually doing this in lab, which can be very frustrating for students. This is really easy once you understand it, but it's a little bit complicated when you're first starting out. All right, so if we look at our X and Y axis first, our Y axis is volume in mils, right? So this is volume of air moved. And this is time, okay? So this would be time in seconds. And so this is pulmonary ventilation, or this is called a spirogram. So what ends up happening, let me explain this first, and then we'll go over how we do this. So this is resting tidal volume. So the arrows indicate the volumes, all right? And these are the typical volumes that we're measuring when we're looking at pulmonary ventilation. So resting tidal volume, all right? How do we do this? You would take a normal breath in, so take a normal breath in, and then let it out. Normal breath in, let it out. Okay, the amount of air you moved is your tidal volume, right? How do we figure out tidal volume? You would take the top of the waveform minus the bottom of the waveform, okay? And usually these are values. Usually this is like 1,000 mils, 2,000 mils, 3,000 mils, 4,000 mils, 5,000 mils, 6,000 mils, all right? And so the average tidal volume, or the average volume that a person can move in a normal breath, is about 500 mils, okay? Then expiratory reserve volume. How do we get that one? Normal breath in, let it out. Normal breath in, let it out. Exhale past that and release. So the amount of air that you exhaled past normal exhalation is your expiratory reserve volume, right? So expiratory reserve volume is a volume of air that you can exhale past normal exhalation. How would you figure this out, or how would you calculate it? It's going to be normal exhalation from your tidal volume minus the exhalation from your expiratory reserve volume, all right? And this is expiratory reserve volume. It's just like the name states. It's the amount of air that you have reserved, or the space you have reserved for maximum exhalation, all right? And then inspiratory reserve volume, which is, the air or the volume you have reserved for inhalation past normal. So normal breath in, let it out. Normal breath in, let it out. Normal breath in, let it out. Normal breath in, inhale past that, and release it. The amount of air you can inhale past normal is your inspiratory reserve volume. So if I was calculating this, I would do the peak of the inspiratory reserve volume minus the peak of the tidal volume, right? And that's going to give you your inspiratory reserve volume. Volumes are actually, how do we figure out volumes? You measure it's a waveform measurement. If we're talking about capacities, like vital capacity is the sum of many waveforms. So notice vital capacity includes your inspiratory reserve, your tidal volume, and your expiratory reserve. So vital capacity is your inspiratory reserve, tidal volume plus expiratory reserve. So that's how much air you can maximally move. So vital capacity is how much air you can maximally move. Notice that this waveform doesn't go to zero. So when we force all the air out, do we get all of the air out of our lungs? The answer is no. What's left over is called your residual volume. So take a look to the left. The total lung capacity includes how much air you can move maximally, so it's your vital capacity, right? Plus your residual volume, how much air is trapped, okay? The alveoli collapse and actually trap air, and so you can't exhale out all the air. So total lung capacity includes both of those measurements. This is a slow vital capacity waveform, you guys. How do I know this? Because we did these measurements slowly. So in lab, you would take one normal breath, inhale maximally, and exhale it all out, right? And then take your measurements. Here are the definitions. So respiratory volumes that we'll investigate in lab, and you should print out the lab and label these so that you know how to measure them. If you were using a spirometry machine clinically, the machine would actually give you the values, but I want you to understand how to measure them. A tidal volume is the amount of air you move in a single breath, about 500 mils. Inspiratory reserve volume is the amount of air you can forcibly take in above tidal volume. It's usually around 3,000 mils or 3 liters, right? Expiratory reserve volume is the amount of air you can forcefully exhale past tidal volume, usually around 1,000 mils. And it's going to depend. It's different for men versus women, and different sizes of bodies. Residual volume is the air that remains in the lungs. It's about 500 mils to 1,000. Your vital capacity is the maximum amount of air that can be moved through the lungs. Vital capacity is your tidal volume plus your inspiratory reserve plus your expiratory reserve, and that's about 4,500 mils or 4.5 liters. Okay, so before we get into forced vital capacity, I wanted to take a second to, let me see, I want to take a second to go over this stuff. Let's see if I can actually do this. Let's go to the whiteboard. Buddy, work with me. There we go. Okay, so here's a whiteboard. Let's move this out of the way. Make this whole thing work. Okay. So if we had to draw this out, let's see if I can get this to look reasonably accurate, somewhat. Okay, so this would be, whoops, this would, too many buttons, you guys, too many buttons. Move this over, there we go. Okay, so this would be my slow vital capacity. So this is your, okay. Here is my graph, right? This would be, let's see, 1 liter, 2 liters, 3 liters, 4 liters. Volume, liters. And then this would be time, right? In seconds. So what you see is when you're doing a slow vital capacity, you're doing a breathing or you're figuring out the vital capacity or the maximum ventilation for a person, but you're doing it slowly. So if we were doing this in lab, we would take a normal breath, right? So inhale, exhale, Inhale, exhale, deep breath in, and then let it out. And then back to baseline. And then we would stop the machine. All right. So this is your tidal volume. That's the normal amount you can move, right? Here's another tidal volume. Here's your inspiratory reserve volume. And here's your expiratory reserve volume. Okay. For the data for the lab, it's a little bit different. It's flipped. And I'm sorry, I know that's a little bit confusing, but for the lab, when they inhale, the waveform goes down. So inhale, exhale, inhale, exhale, inhale maximally, and exhale maximally, and back to baseline. So it's, whoops, so it's, stop. So it's flipped from what we would get in lab. Okay, so just make sure that you pay attention to when you're analyzing the data for lab. All right, let me pick a different color for you guys. Let's see, where's my, pop up over here. Give me a second to putz around with this. Select. Whiteboard. Maybe format. There we go. Okay. Let me see what color is going to stick out. Let's try the blue and see how that works. Okay, so for lab, you're going to calculate or analyze or figure out the tidal volume. So the tidal volume, what you're calculating is you're figuring out the peak minus the trough. So you need to extrapolate what is this number right here, and you would call that, let's say it's 2.5. Okay, it's a bad drawing, but we'll call this 2.5 liters. And then this would be at 2 liters. So what is your tidal volume? So your tidal volume is 2.5, the peak minus the trough, which is 2.0. So your tidal volume should be 0.5 liters, okay, or 500 mils. How would you calculate the IRV then? So remember, inspiratory reserve is how much you can inhale past normal. So normal was the peak of your tidal volume, which was 2.5, right? And then your inspiratory reserve, the peak of that, let's say this was 3 liters. Okay, now it's not accurate, obviously, but the spirogram that you're using for lab will be accurate. So your inspiratory reserve is going to be 3 minus 2.5. And so this is going to equal, again, 0.5 liters, right? That's your inspiratory reserve. When you are doing these for lab, I want you to print out the data, and I want you to actually label it and do your calculations and show your calculations on your slow vital capacity data, okay, on the actual spirograms. How would you calculate your ERV? So your expiratory reserve volume is the amount of air that you can exhale past normal exhalation. So normal exhalation was shown or measured in your tidal volume. So we're gonna extrapolate this over. Okay, so normal exhalation is 2.0 liters. And then where does the bottom of this waveform end? Okay, we're gonna say it ends at 1 liter. So this is 1.0 liters. So expiratory reserve volume is 2 minus 1 liter. So that would give us 1.0 liters. Okay. What else can we calculate from this? We can add these guys up, and that would be your vital capacity. So inspiratory reserve plus tidal volume plus expiratory reserve is your vital capacity. So let me see if I can get another color. Let's do orange. Okay. So your inspiratory reserve, I'll circle it, right? Plus your tidal volume, plus your expiratory reserve is going to be what? Your vital capacity. So vital capacity is inspiratory reserve plus tidal volume plus expiratory reserve volume. And that should equal, or if you look at it, inspiratory reserve was 0.5 liters, tidal volume was 0.5, and then ERV was 1.0. So that would be around 2 liters. And of course, this is way off, but that's okay. What else can we measure from that? So keep in mind, again, this was done slowly. So this is your slow vital capacity. The other thing that you can get from this is your residual volume. So let's change the color. Your residual volume is how much air is trapped, remember, because this doesn't go down to 0. So this would be your residual volume, right? Residual volume is whatever the bottom of your ERV is, subtract 0 from that. In our waveforms in class and on the data, I think on the data sheet, the data that I got for you, residual volume, you have to calculate it because you can't measure it off of there. Some of the spirograms actually cut, they put the bottom of the waveform at 0, so you can't measure it. So you have to calculate it. And I gave you the equation how to calculate it on the handout if you need to. So if we want to include the residual volume in the measurement, it is going to be your slow vital capacity plus residual volume. That will give you your tidal volume. So your tidal volume is your slow vital capacity plus your residual volume. Okay? Or no, sorry. Your total, that's wrong. Total lung capacity is your slow vital capacity plus your residual volume. So total lung capacity is how much air you can maximally move plus the stuff that's trapped, the residual volume. Okay, let's go back and cover forced vital capacity then. And then I'm going to draw it out for you as well. See if I can save it. Let's see. Whoops. We're going to switch to the PowerPoint. Okay, so forced vital capacity is measuring your vital capacity when you forcefully exhale, right? And again, you're measuring vital capacity. So theoretically, this should be the same as slow vital capacity. You're just measuring it forcefully. So, excuse me, forced vital capacity is measuring your vital capacity when you forcefully exhale. This is what they would do, or this is what the clinician would do if you were having breathing issues. So you would come in, they would make you do a breathing test, and this is what they're actually looking for. So they would make you take a deep breath in and then force it all out in 3 seconds, as hard and as fast as you can. And if it didn't look like this curve in the picture, then they would make you redo it a couple of times. What are they looking for? They're looking to see if you have a pulmonary obstruction or a pulmonary restriction, or if your breathing test came back normal. So FVC stands for forced vital capacity. FEV1 stands for forced expiratory volume at 1 second. So how much air did you forcefully exhale in 1 second, right? So let's take a look at this. So again, remember your units. This is volume in liters. We can assume that that's 1 liter, 2 liters, 3 liters, 4 liters. And this is seconds. So 1 second, 2 seconds, 3 seconds, 4 seconds. You take a deep breath in. If you did this in lab, you take a maximal breath in using all of your muscles and then force it out as hard and as fast as you could. The peak of this waveform would actually be your vital capacity. So the amount of air you can maximally force out would be the peak of this. And you want to know how much air you moved in 1 second, right? So you want to know, okay, what was the measurement, or how many liters did I exhale out in 1 second? And how many liters did I maximally exhale out, right? And again, this is forced vital capacity, not slow vital capacity, because you're forcing it out in 3 seconds. We can use a forced vital capacity to figure out if somebody has a respiratory disease, okay? So FVC, again, a little bit redundant, but I can't stress this enough because it's hard for students to understand how to do this. And it's equally hard because we're not face-to-face doing this in lab. So it's measuring your vital capacity when you forcefully exhale. So when you go in for respiratory tests, or if we did this for lab, we would put in a person's parameters. So age, weight, height, gender, and ethnicity. And that would give us predicted values. All right. And so we would come up with, the computer has an algorithm that would come up with your predicted vital capacity. And so then we would blow into the tube, and what you want to see is that the actual force vital capacity, how much you actually forcefully blew or moved, would be greater than or equal to predicted, what you should have blown, okay? So how much air you maximally moved should have been greater than what was predicted for you based on your age, weight, height, ethnicity, and gender, okay? If your actual forced vital capacity is less than predicted, that shows that you have a restrictive pulmonary disease, right? So that's one thing. So make a note that this shows a restriction. Let me see if I can get this. Right? So if actual is less than predicted, then you have a restrictive pulmonary disease, right? What does that mean? So actual forced vital capacity, let's do your forced vital capacity again. So deep breath in, do it with me, and then blow it all out as hard and as fast as you can, okay? Let me change it to video so I can actually show you. Sorry, guys. So deep breath in, right? Blow it all out as hard and as fast as you can. That is your actual forced vital capacity, right? Deep breath in, blow it out as hard and as fast as you can. Okay, that's your actual forced vital capacity. That should be equal to or greater than what's predicted based on your parameters, right? If somebody has a restrictive pulmonary disease, that means that they have some type of damage to the lung lining. Okay, so think of those alveolar sacs. That's damaged. So let's say somebody had pneumonia, a bacterial pneumonia that damaged the lung lining, right? Or the coronavirus that damaged the lung lining. Tuberculosis or asbestos or black lung disease or cancer, right? So a restrictive pulmonary disease means that the lung lining is restricting ventilation or restricting expansion, okay? So how do I explain this to students? Think about having a brick on your chest, right? Or an elephant on your chest. So if something's on your chest, you can't take a maximum breath, okay, or a maximal breath like you could. So normal, deep breath in, force it all out. When you have a restriction, you can't inhale and move as much air as normal. So deep breath in, blow it all out. Deep breath in, blow it out. Okay, the lung lining can't expand and recoil like it would in a normal healthy individual. And so normally you can do this, right? With ventilation, if you have a restriction, you might be able to move it this much, right? So it's going to be way less. Your actual is going to be way less than predicted. Okay. For the second portion of this, normally your forced expiratory volume, so if you look at the next one, forced expiratory volume 1 divided by forced vital capacity times 100 should equal 80% or greater in a healthy individual. So what this means is in 1 second, you should be able to forcefully exhale out 80% of maximum if you're a normal, healthy individual. If your percent FEV1 is less than 80%, it shows an obstructive pulmonary disease, right? So if FEV1% is less than 80, it shows an obstruction. So let's demonstrate that. So deep breath in, force it all out. This is normal. Deep breath in, force it all out. That's normal. Okay. Now, deep breath in, blow through a boba straw. Really easy. Deep breath in, blow through a McDonald's coffee straw. Really, really hard. Okay. That is what it's like when you have an obstructive pulmonary disease. Okay. The bronchi are constricted. The trachea is constricted. It's obstructing air from flowing through those tubes. And so you can't, normally you'd be able to get out 80% of your maximum by forcefully exhaling. When you have an obstruction, you can't get out 80% of max because those bronchi or trachea are too inflamed or constricted. And so this would be an example, or an example of this would be, asthma, bronchitis, something that's going to affect the trachea and the bronchi. This does not mean that if you have a restriction or an obstruction that they can't fit into both categories. So chronic obstructive pulmonary disease starts as an obstruction, right? But then it can lead into a restriction. So for people who have emphysema, that is damaging the lung, cat's trying to jump on my computer. For emphysema, what you see is it's obstructive first, so you get inflammation from chronic cigarette smoking and excess mucus and debris and damage to the lung lining. But then as the emphysema progresses, you're going to lose that elastic recoil of the lungs, and so you lose the surface area ability of those lungs. And so it starts out as an obstruction and then turns into a restriction. Okay. So let's see. So if, if your forced vital capacity, your actual forced vital capacity is less than predicted, that is evidence of a restrictive pulmonary disease. That means that alveolar surface area is diminished. You have damaged that alveolar surface area, you form scar tissue, and you don't have enough gas exchange surface area. So this could be from tuberculosis, pneumonia, some type of fibrosis. Note that myasthenia gravis also falls under this category. I want you to think, why does myasthenia gravis fall under this category? So what is myasthenia gravis? All right, so your body forms antibodies against your nicotinic cholinergic receptors, right? And that is going to affect nerve-to-nerve communication and nerve-to-skeletal muscle communication. What's the major skeletal muscle that's involved with breathing? The diaphragm, right? So if you have myasthenia gravis and your antibodies start knocking out your nicotinic cholinergic receptors for your diaphragm, are you going to be able to move as much air as you normally would? No, you're not. So your actual forced vital capacity is going to be less than your predicted. If percent F, this should be FEV1. So let me change that. If your percent, I'm going to line through this, FEV1 is less than 80%, that shows evidence of an obstructive pulmonary disease, right? What does this mean? An obstructive pulmonary disease means you're obstructing air flow through those tubes. So the bronchi are constricted or obstructed. Again, early stages of emphysema will show this. Chronic obstructive pulmonary disease, one of them is emphysema. Asthma, right? Bronchitis, lung cancer, pulmonary edema, decreased left heart output, lung infections, right? So what are we seeing with the coronavirus? You're actually seeing a restrictive respiratory disease. And so people who are recovering from the coronavirus may have long-term damage or scar tissue because of the virus attacking the lung lining. Here's a plot. This is actually really interesting. I want you to draw out the forced vital capacity plot and talk your way through it, right? So take a look. So the red line is normal lungs. So you're going to take a deep breath in, you force it all out, and you try to force it all out within 3 seconds, okay? This is volume exhaled in liters. This is time in seconds, okay? Take a look. We're interested in the 1 second reading. So the maximum, if you look at the red line, the maximum value is your forced vital capacity, right? So the maximum value is always going to be your forced vital capacity on these graphs. And then you want to know how much air you moved out in 1 second compared to max, right? This should be 80% of max. So you should have you should have exhaled out 80% of your maximum amount in 1 second. That's normal. Let's look at obstructive diseases. Okay, so this is the blue line or the green line. So you take a deep breath in, you force it all out. Okay, this is how much you forced out in 1 second. This is how much you got maximally. Do you think you're forcing 80% out in 1 second? You're actually not, and you could do the calculations. So you can't get out 80% of maximum in 1 second. Why? Because the bronchi are obstructing airflow. The bronchi are constricted, and so they're obstructing getting that getting all that air out in 1 second. Think about breathing through a McDonald's coffee straw. Will you exhale out all of the air that's in your lungs, or that you can maximally exhale out? Yes, you would. You just can't do it in 1 second because the bronchi are preventing that. Okay. Take a look at a restricted disease. So this is the yellow line. So the yellow line, notice deep breath in, you're forcing it all out. Notice that you can get out 80% of max, but look at your maximum. Your maximum is way lower, okay? And both of these max are gonna be way lower, but for a restrictive disease, you really can't get out much. Your forced vital capacity is way lower than normal. So if you did this percentage, this may be normal, but the amount of air that you can get out maximally is way less. So your actual is way less than what's predicted for you. For an obstructive disease, this is way less than 80% of that. Okay, this is going to take some time to get through for you to understand this. It's not easy, but it will help you when you get into your disease states for your respiratory system in your professional programs. Let me see. I know there's a way to go to the next one. Let me see. Oh, here we go. Okay, so let's do a forced vital capacity plot. So here's your forced vital capacity, right? Here's your Y, your X. So let's say this is 1, 2, 3, 4, 5 liters. So this is volume in liters. And this would be seconds, right? Whoops, time in seconds. It's actually recent. Good thing the eraser button works. Here we go. Okay, let's try this again. Text. Nope. I want it black. Okay. So this would be time. Oh, good Lord. Draw. Whatever. That's fine. time in seconds. Okay. Okay, so to do forced vital capacity, if we were doing this in lab, you take a deep breath in and force it all out in 3 seconds. Okay, so what does that look like? Deep breath in and then force it all out in 3 seconds. And eventually you would plateau and it would look something like that. Okay. This would be, let's see, this would be your 1 second mark, 2 seconds, 3 seconds, and whatever this is, 4 seconds. Okay. What are we interested in? We're interested in how much air you moved out in 1 second, and we're interested in how much air you moved out maximally. Okay, so let's go back to the blue marker. Whatever the peak number is, that's going to be your forced vital capacity. Okay. And then we want to know how much air you moved out at 1 second. So you would go up to your line you blew, and then you would go over to the volume and you would figure out the number. So this is 1 liter, 2 liters, 2.5 liters. Okay. So my forced expiratory volume 1 is 2.5 liters, right? What is my forced vital capacity? You'd extrapolate over, that looks like that is 3.5 liters. Okay, so let me change the colors. So your percent FeV1 would be 2.5 liters divided by 3.5 liters times 100. Okay, and that would give you your percent FeV1. So this is the test you would use to figure out if you have an obstruction. For a restriction, you're looking at your actual forced vital capacity. So how much air did you move? So actual forced vital capacity is 3.5 liters. And then what is your predicted based on your age, weight, height, and gender and ethnicity? If your predicted forced vital capacity, let's say it was 3.2, does this show evidence of a restriction? No, it doesn't, because your actual is greater than or equal to your predicted. What if my predicted forced vital capacity was, oops, what if my predicted forced vital capacity was 4.5 liters? Does that show evidence of a restriction? It does, because the predicted is higher than the actual is way less. So that shows evidence of a restriction. So this is what you would use clinically to figure out if somebody has a restriction or an obstruction. Okay. For lab, what you're going to do is you're going to, let me go on to the next one. For lab, remember the waveforms are flipped. I apologize for that. It was the only good one that I could find. So what they did is instead of going up, the waveform actually goes down. So they took a deep breath in and then, or deep breath in, and then they forced it out. And so what you see for this one for lab is the curve is inverse, okay? You can still figure out the values, it's just flipped a little bit. So this is still gonna be your 1 and this is still gonna be your max, right? Just pay attention to your measurements or your units for your X and Y axis. And if you have any questions, let me know. Okay, let's move on. So I'm going to go back to the PowerPoint. Let me save this. There's my save button. Let's see. Okay. Let's get into gas exchange. So gas exchange is respiration. It occurs across the respiratory membranes, meaning the alveolar wall and the capillary wall. Remember, gases diffuse from high concentration to low concentration. So that's going to be our motif for the next couple of weeks. Your alveoli are microscopic membranous air sacs. They're comprised of simple squamous epithelial tissue. They're the functional unit of lungs and the functional unit of gas exchange between your lungs and your circulatory system. And of course, gas exchange occurs by diffusion. Surfactant cells produce surfactant that's going to reduce surface tension so that the alveoli don't collapse. So it reduces friction. Here is the alveolar sacs, right? And notice the alveolar sac, they're made up of clusters. It's like a cluster of grapes, right? So a cluster of alveoli. Each one is called an alveolus. Here's your pulmonary artery, or actually it's a capillary bed, but pulmonary artery tributary, and then pulmonary vein tributary. Low oxygenated blood comes in, grabs oxygen, kicks out CO2, and then the oxygenated blood's going to go back to the heart to get kicked out to the body. Keep in mind, if you look at the top picture, diaphragm, right? Look at the smooth muscle wrapped around the bronchioles. These are going to constrict and dilate. So if somebody has asthma and they're having asthma issues, this smooth muscle actually constricts and constricts or prevents the air from moving. So that provides an obstruction. We can skip this one. It's just a picture of the alveoli. Gas exchange. So let's take a second to look at this. Now, you know that here's the alveolus, and then here's a capillary, right? You know that oxygen is higher at the alveolus, and it's going to go into the capillary. And that CO2 is higher in the capillary, it's going to get kicked out into the alveolus. But I want you to look at the numbers. So this is oxygen level. So you may think that this is pressure of oxygen, but you may think that this is percent, but it's actually not percent. It's in, I believe it's in torr. Sorry, I totally forgot my train of thought. And so the amount of oxygen coming in is 40 units. CO2 is 45. At the lungs, oxygen is 100. CO2 is 40. And so when the blood cells, or when the blood comes to the area of gas exchange in the alveolus, oxygen is going to go from where it's highly concentrated to where it's least concentrated. So it's 100 at the alveolus, it's 40 at the capillary, so oxygen is going to move in. CO2 is going to move down its concentration gradient as well, or it's, since they're gases, it's actually partial pressure, but CO2 is going to move from where it's highly concentrated to where it's least concentrated, and so CO2 is going to move down its concentration gradient as well. So leaving the capillary, we have 100 units of oxygen and 40 units of CO2. Look at the difference in oxygen and look at the difference in CO2. So oxygen, 100 versus 40. CO2 is 40 versus 45. Which one changes drastically and which one doesn't change very much? Oxygen changes drastically, CO2 doesn't change very much. Your body regulates CO2 very heavily. And the evidence for this is that there's not much flux in CO2 levels. CO2 levels are going to be around 40 to 45 units, whether we're talking about arterial blood versus venous blood. And so this suggests to me that it's highly regulated. Another thing too, when we get into regulation, is there's actually 2 monitoring stations for CO2. You have a peripheral monitoring station in your aortic arch and carotid sinus, and then you have a central monitoring station in your medulla oblongata. And so why do we regulate CO2 so much? Or why do we have so many monitoring stations of CO2? Why don't we want it to shift very much? Excess CO2 produces what? So excess CO2 in water is going to produce acid. And when you produce acid, if this blood acidifies, what happens to the hemoglobin? As the pH drops, the hemoglobin is going to have, it's going to lose its affinity for oxygen. So it's going to make it harder for oxygen to bind to the hemoglobin. Okay, and so it's really regulated. Take a look at, this would be your interstitium, and these would be your cells, right? So here's internal respiration. As the blood is coming through the capillary bed, your oxygen level is 95. At the tissues, it's 40. Why is it so low? Because the tissues have been using oxygen for metabolism, right? So they use it at the electron transport chain at complex 4, which you just learned. And they are producing CO2 at the molecular level. So oxygen is higher in the capillary bed. It's going to go into the tissues, and then CO2 is higher at the tissues because you're making CO2 because of metabolism, and so it's going to move out into the bloodstream. Notice that excess CO2, what happens when we have excess CO2? It's going to acidify the tissues, and that's going to acidify this area. Okay, so when you acidify this area, and this area is also warm, remember, because it's near the tissues. They're actively respiring tissues. So when you have acidic blood and you have a higher temperature, it's going to favor oxygen getting dumped off of the hemoglobin, which enhances oxygen delivery to the tissues. Okay, take a look at oxygen. So oxygen levels 95 versus 40 leaving the capillary bed. CO2 40 versus 45. Again, you don't see much of a shift in CO2 because your body regulates that CO2 heavily. Okay, so I'd like you to know how they're transported in the blood, how the gases are transported in the blood. And so here's the capillary on the top left, lung, then your red blood cell, and then your tissues on the bottom right. So oxygen diffuses into the plasma. Some of it stays in the plasma, not much, about 2%. Most of the oxygen goes into the red blood cell and binds to hemoglobin. So most of your oxygen, it gets transported in the blood bound to oxygen, right? Or excuse me, bound to hemoglobin. So that's 98%. So think about, how do we measure this for a person? So we're going to take a pulse oximeter and we're going to measure the percent saturation, right? So percent saturation of hemoglobin with oxygen, and normal is anything above 90, usually 97 to 100% hemoglobin saturated with oxygen. Okay, what happens when we get to the cells? The oxygen is going to dissociate from the hemoglobin, right? Because there's less oxygen here, there's more oxygen here, so oxygen is going to go into the cells. Plus, this has a higher temperature, and this has a more acidic pH, and so it's going to favor hemoglobin releasing oxygen, right? Good. Oxygen transport's really easy, okay? And this just covers a temperature curve, and we've already covered that in blood, so go back and look at your blood lecture. The effective pH on hemoglobin saturation. So again, to reiterate, the Bohr effect is the effect pH has on hemoglobin saturation curve because of CO2. When there's excess CO2, because CO2 and water make acid, you have excess protons, right? So the CO2 plus water in the presence of carbonic anhydrase, what would happen regardless? But carbonic anhydrase is that enzyme on the red blood cells that catalyzes this reaction to make carbonic acid, and then that dissociates into protons and bicarbonate ions. So let's take a look at this. And again, this is pH effects on hemoglobin, so you should take a look at the blood lecture again, because we talked about it already. But how is CO2 transported in the blood? 70% is transported as carbonic acid, but we're not done. Once it's formed, it separates into H plus and bicarbonate ions. And we need to talk about the chloride ion shift. Some of it is transported via hemoglobin, and then some of it is transported in the plasma. Let's take a look at this. So CO2 is generated at the tissues, right? Through cellular respiration. So CO2 is produced in the tissues. Some of it is transported in the plasma. A lot of it goes into the red blood cell, right? Where it binds with hemoglobin to make carbaminohemoglobin. That's 23%. But then most of it binds with water in the presence of carbonic anhydrase to make carbonic acid. As soon as that's made, it splits into bicarbonate ion and protons. The protons are actually going to be bound to hemoglobin. The bicarbonate ion gets kicked out of the red blood cell. But if you are taking a negative, if you're kicking a negative out, you have to exchange it for a negative because this is neutral inside the cell. So this is called a chloride ion shift. It's just the process of kicking the bicarbonate out, which is negatively charged, and you have to exchange it with a negative ion, which is the chloride ion. So most of your CO2 is transported in the blood as bicarbonate ion. Keep in mind that bicarbonate ion is actually going to act as a buffer. So let me change this to pen. Let's do black. Oops. Okay, so this bicarbonate, it's a buffer, right? So what does a buffer do? Does a buffer keep the pH at 7, or does it keep the pH neutral? It's going to keep the excuse me. Let me rephrase that. So is a buffer going to keep the pH at 7, or is it going to keep the pH of the solution, whatever pH it is? All right, so the buffer maintains a pH, right? Right? What is the what is the pH of your blood? The pH of your blood is 7.35 to 7.45. All right, so bicarbonate ions are one buffer that we use to keep pH at 7.35, 7.45. How does it work? Bicarbonate, we'll draw albumins too, and I think I talked about this. Here's our albumins are what are negative plasma proteins. They're big negative plasma proteins. So these both, in addition to our phosphate buffering system, let's see, let's do red. So when we have, let's say we're lifting weights or we're exercising, or for whatever reason we're making, we're increasing the metabolic production of H+ of protons, okay? Let's see if we can go back. Oh, you guys, I'm so sorry. I just lost it all. Anyway, so the point of it is that your bicarbonate is going to grab the free H+ so that it's no longer free. So it's bound to the bicarbonate. Your albumins do the same thing. So albumins are going to grab the free H+ so that it's not free and it's not doing damage. What shifts the pH? It's actually the free protons floating around. And so if you bind it to bicarbonate, or if you bind it to albumins or your phosphate groups, then it's not going to damage your body tissues and your cells. Okay. Oh, okay. There you go. So what happens when you transport it to the lungs? The CO2, so there's less CO2 at the lungs, so all of the CO2 is going to get kicked out. All of the CO2 is going to go from where it's highly concentrated to where it's least concentrated. The CO2 bound to the hemoglobin is going to dissociate and go into the alveolus, right? And then the bicarbonate that's in the plasma is going to go, again, it exchanges with chloride. It's going to go into the red blood cell, reform carbonic acid, and then go through carbonic anhydrase to make water and CO2. And so the CO2 is ultimately going to get kicked out of the lungs. I would like you to know this process in detail. It's important to know how CO2, now that we know from respiration or cellular metabolism how CO2 is made, we want to know how it's transported in the blood and then how it's kicked out, okay? We also need to know how it's regulated, right? So how do we regulate ventilation? First, let me say that your primary mechanism to breathe is based off of CO2. Excuse me, it's CO2 levels in the blood. CO2 increases, that triggers your respiratory center and medulla oblongata. It causes you to take a breath. CO2 increases, you take a breath, CO2 drops. CO2 increases, you take a breath, it drops, right? So respiratory neurons in the medulla oblongata control inspiration and expiration. Neurons in the pons modulate ventilation, so they oversee it. Rhythmic pattern of breathing arises from a network of spontaneously discharging neurons. Now, you know from last when we talked about the nervous systems that because this is skeletal muscle regulated, it is somatic nervous system, right? And somatic nervous system is voluntary. So it's nerve synapsing with skeletal muscle, and that nerve tells that skeletal muscle to contract, or your brain tells that skeletal muscle to contract, right? And so this is voluntary. So normally, if you think about your biceps brachii, when you want to contract it, you have to tell your brain has to tell your biceps to contract, right? It's the same thing with your respiratory muscles. You're just not as aware of it. So you can, so it's not really involuntary, but it's, you don't have to consciously regulate breathing, but if you want to consciously regulate breathing, you can, right? And so for lab, I want you guys to do normal breathing and then exhale and hold your breath and time how long you can hold it. Then I want you to hyperventilate and then exhale it out and hold it. And then I want you to breathe into a paper bag or a plastic bag, exhale and hold it, and see what the difference is. So what you're doing is you're playing with CO2 levels and you're seeing how that is regulating breathing, right? Or how long you can hold your breath based off of that. What you'll see is as CO2 accumulates, that's going to trigger you to take a breath. And you can practice that now. So you can take a breath, right? Normal breath, exhale, and then hold it. And as you're holding it, think about how that feels and hold it as long as you can. So as you're holding your breath, what happens? You start to get more anxious, right? You start to feel like you can't breathe. You start to feel panicky. Okay, that's because CO2 is building and building and building, and it's triggering your chemoreceptors. Your CO2 is triggering your chemoreceptors as it builds, and as acid builds, because CO2 builds, it's telling your brain to take a breath. And eventually, you're going to have to take a breath, right? So you're forced to take a breath. You can try holding your breath for as long as you can. Children, if a child's mad, they'll hold their breath and they'll try to hold their breath as long as they can, thinking that they're going to die, and they can't because they're forced to take a breath and they get more angry or they get more irritated, right? But your medulla oblongata does this as a way to regulate it. So if you aren't breathing properly, or if you're hyperventilating or whatnot, your medulla oblongata is going to knock you out and regulate it for you. So it's going to oversee breathing. Okay, so this just shows, your inspiratory chatter, nervous chatter. So nerves increase firing, and that causes your diaphragm and external intercostals to start contracting, and you take a breath in, and then those nerves shut off, and because they shut off, the muscles relax and you exhale out, and then the muscles go, the nerves chatter, get louder, take a breath in, and then the nerves shut off, the muscles relax, and then you exhale. So inhalation is active, right? Because you're contracting those muscles, those nerves are on. And then exhalation is passive. Those um, nerves shut off the skeletal muscle and the skeletal muscles relax. For regulation of ventilation, there's 5 sensory modifiers of your respiratory centers. The big one that I want you to know, obviously, is your chemoreceptor reflex. So chemoreceptors are sensitive to CO2 levels and oxygen levels or pH of blood. Chemoreceptors are your primary mechanism to breathe is based off of CO2. So as CO2 goes up, the amount of protons go up and the pH drops. So as CO2 goes up, you make more acid. Okay, those chemoreceptors are sensitive to CO2 and proton levels or acid first, and then their secondary mechanism is oxygen. So a lot of students think that your primary mechanism to breathe is based off of less oxygen or low oxygen in the blood, and it isn't. It's based off of CO2, because remember, we keep CO2 within a tight range because it affects pH greatly. Secondary to that are baroreceptors. Obviously, if your blood pressure drops, you're going to kick up breathing to sort of help make sure you get oxygenation. Stretch receptors, irritating physical or chemical stimuli, and various other sensations. You know that when you're in pain, ventilation increases, changes in body temperature, ventilation increases as well. Right, so let's take a look at the chemoreceptor reflex. So for lab, I want you to diagram this out, and I want you to know this for lecture and lab. This is really important. All right, so your chemoreceptor reflex, let's say you have an increase in CO2, right? So your mechanism to breathe is based off an increase in CO2. And if CO2 increases, what happens to pH, right? So we have so the stimulus would be an increase in CO2. And if we have an increase in CO2, what happens to the amount of protons, right? So you're going to make excess protons, and what happens to pH? pH drops. Is that acidic or basic? Good, it's acidic, okay? So that's your stimulus. Where are your sensors? You have 2 of them. Central chemoreceptors are the first sensor, so make sure you're drawing this out. They're located in the medulla oblongata. The second sensor, or the group of sensors, are peripheral chemoreceptors. They're located in your carotid and aortic arteries, actually in your carotid sinus and aortic arch. Okay, so those are your chemoreceptors. Those chemoreceptors are going to send a signal where? So your chemoreceptors, oops, sorry. So your chemoreceptors, and I want you to know both sets for the exams, are going to be your sensors, and then they're going to send a signal where? So what's your integrator? Your integrator would be your medulla oblongata, right? And that's going to measure it against your set point. And medulla oblongata is going to say, hey, CO2 is too high, or pH is too low, or oxygen is too low. I'm going to send a signal out to what? Good. So your respiratory muscles. So the effectors are actually your respiratory muscles. Can you name one? Good. Your diaphragm and your external intercostals, your sternocleidomastoid, your scalenes. Good. And your pecs. And so those respiratory muscles, what's going to happen? So you're actually going to increase your respiratory rate and your depth of breathing. And so that should decrease your CO2, okay? The chemoreceptors are a little bit different in how they function. So the central chemoreceptors are in the medulla oblongata. They detect your interstitial fluid CO2 and your pH. They're going to monitor arterial CO2, cerebral blood flow, and metabolism. Your peripheral chemoreceptors detect arterial CO2, pH, and drop in oxygen. So these ones are your peripheral chemoreceptors are also sensitive to a drop in oxygen, and these guys are going to monitor alveolar ventilation. Let's take a look side by side. So increase in CO2 is your stimulus. If we go in the middle, that's going to increase CO2 of your artery. Increased CO2 is going to increase protons. That's going to drop the pH and increase bicarb. That increase in protons and increase in CO2 is going to stimulate your peripheral chemoreceptors. That's going to cause you to increase ventilation. Okay. Look at the left side. So at the top, increase in CO2 increases CO2 of cerebrospinal fluid. Increase CO2 of cerebrospinal fluid plus increase protons in your cerebrospinal fluid, or an increase in protons is going to stimulate your central chemoreceptors. That's going to increase ventilation. Okay, so that is your primary mechanism to breathe. Primary mechanism is increase in CO2, which causes an increase in H plus or protons. An additional, you could increase protons, which would be a metabolic way or a metabolic issue, like let's say diabetic ketoacidosis. So you're making excessive ketones, it's acidifying the blood. You're not doing anything with CO2, but you have acidic blood. And so is that going to increase ventilation or decrease ventilation? It's going to trigger your chemoreceptors and you're going to increase ventilation. Okay, so that would be some type of pathological issue. Let's take a look though at low oxygen levels. So if you have low oxygen levels less than 60 millimeters of mercury, that's also going to stimulate your peripheral chemoreceptors for you to take a breath. Okay, so again, this isn't your primary mechanism to breathe, it's your secondary mechanism to breathe. So let's say, I go up to Mammoth where, the altitude 12,000 feet, there's less oxygen. That is also going to stimulate my peripheral chemoreceptors to increase ventilation as well. So that's how we would use that. I like this picture because it labels the stimuli, sensory receptors, your afferent neuron, integrating center, which is the medulla, efferent, and effectors. And so it has number labeled. So you can take a second to look at this. Inspiration is on your left, expiration is on your right. Let's end then with alveolar ventilation and CO2 levels. So letter B is normal. So as CO2 increases, alveolar ventilation increases, right? And this is with normal oxygen levels. So this is 85 millimeters of mercury. So the units are millimeters of mercury. Take a look at when we have less oxygen. So when oxygen levels are diminished, less than 60, what do you see? As CO2 increases, ventilation increases more than normal, right? So the slope is steeper. So when there's less oxygen and you have increase in CO2, you're going to do more ventilation. Take a look at what happens when you have normal oxygen and you've drank 7 beers. So when you drink alcohol, that actually depresses your central nervous system. So take a look that as CO2 levels increase, look at how low your alveolar ventilation is. It actually diminishes that reflex. So alveolar ventilation is low compared to what it should be normally. Okay, go ahead and get started on the lab and email me if there's any questions. Hopefully this should make the lab easier. I'll talk to you soon. Oh, last thing, sorry. This is, and I don't like focusing on this, but I want you to know that ventilation is based off of CO2 and oxygen. But when blood pressure drops, it's sort of a secondary mechanism that when blood pressure drops, ventilation is going to kick up. Okay. So I want you to think of when your respiratory chemoreceptors are stimulated, you're going to increase depth and rate of respiration. So why do you hold your breath longer when you hyperventilate? Okay, so when you're hyperventilating, if you're breathing and forcing all that air out, you're actually kicking out more CO2 than you normally would. And so it's going to take longer for you to build up CO2 to trigger those chemoreceptors to take a breath. Okay, why can you not hold your breath as long when you breathe into a bag? right? That's what I want you to focus on for the first page of the lab exam, or the lab. With chronic obstructive pulmonary disease, so let's cover COPD. So chronic obstructive pulmonary disease means that they have damage of the lung lining, so they can't get out the CO2. So they have an accumulation of CO2 over time in their plasma because they have that damaged alveolar surface area. So over the decades, right, over the years and years and years, CO2 slowly increases. As CO2 slowly increases, it's not going to trigger the respiratory centers, or your chemoreceptors don't respond to that. Chemoreceptors respond to a drop in CO2 or an increase in CO2 relatively quickly. So what happens? Those chemoreceptors have a higher set point for CO2, right? So our set point in a healthy individual is much lower than someone who has chronic obstructive pulmonary disease. So their set point for CO2 is higher. So because CO2 is high and it stays high, your chemoreceptors aren't triggered, or this person's chemoreceptors aren't triggered to take a breath. So what becomes your driving force for breathing? It's actually a drop in oxygen. So that drop in oxygen is now stimulus to take a breath, right? So what happens when you give them oxygen, right? So if you give somebody who has chronic obstructive pulmonary disease, severe chronic obstructive pulmonary disease, oxygen, is their oxygen level ever going to drop low enough that it triggers them to take a breath? No, right? So you have to be careful when you give them oxygen. You don't want to give them too much oxygen. You want to do 1 liter, 2 liters instead of, 8, 9, 10, because you need that oxygen level to drop still so that they remember to take a breath. Okay, good. We're done for real this time.